Hello grade 12. So after the lack operon concepts, today we will discuss about Human Genome Project. Okay? If we are talking about silent feature of the Human Genome Project, that is ambitious 13th-year project that launched internationally to determine the base pair that comprising the human DNA. And this project, which saw completion in 2003, was also aimed at identifying and mapping all the 20,500 genes of human genome, not only from the physical standpoint but also from the functional perspective. Okay, so today we will discuss about all these points in lecture. So let's start today's lecture. So these are the content that we will discuss in today's lecture. First, Human Genome Project, which consists silent feature of human genome. Next, what was the Human Genome Project, milestone and goals of HGP, issues of concern, future challenges, and the last topic that we will discuss, factors for large-scale genome project, which include YAC and BAC. YAC is basically yeast artificial chromosomes and BAC is bacterial artificial chromosome. And the last is the difference between YAC and BAC. But in the today's lecture, we will discuss about silent feature of Human Genome Project, and second, what was the Human Genome Project. Okay, so Human Genome Project, first of all, we have to know that what is human genome. Human genome is the complete set of genetic information for human. The scientific name of human is Homo sapiens. And the human genome is by far the most complex and largest genome. And the lifespan, that a length of about 6 feet of the DNA, that means containing more than 30,000 genes. The DNA material is organized into a haploid chromosomal set of 22 autosomes. There are 23 pairs of the chromosomes, so out of these 23 pairs of chromosome, 22 pairs of chromosome are autosomes and one pair is the sex chromosome, that is differ in male and the female. If we are talking about the male, so there is Y, and if we are talking about the male, then female, that is X. Okay, so in this slide you can see human genome sequencing that is done by 2/11/2001, in which you can see 22 autosomes are there plus 2 sex chromosomes are there, X and Y. Y is the smaller one. Now the silent features of human genome. Human genome basically consists the information of 24 chromosomes. I told you in 24 chromosomes, 22 pairs of autosomes and one is X chromosome and second one is Y chromosome if you are talking about the male. In Homo sapiens, that means 2n. 2n, n is the number of the chromosome. So 2 multiplied by 23, that means 46 chromosomes are there. Human genome basically consists or contain over 3 billion nucleotide base pair. That in 11th class you have already studied about nucleotides and nucleosides. Human genome is estimated to have about 30,000 genes. So average gene consists of 3,000 bases. But the size of gene that vary greatly, with the largest known human gene that encoding dystrophin, which consists 2.5 million base pair. Only about 3% of the genome which encodes amino acid sequence of polypeptide, and the rest of is the junk, that means repetitive DNA. and the functions are unknown for over 50% of the discovered gene. Now, the repetitive sequence that I told that is junk, so that make up very large portion of the human genome. Repetitive sequence have no direct coding function, but they shed light on the chromosome structure, dynamics and evolution. Chromosome 1 that has most gene 2968 and Y that has the lowest 231 gene. So almost all the nucleotide bases are exactly the same in all peoples. Genome sequences of different individuals that differ for less than 0.2% of base pair. Most of these differences occur in the form of single base difference in a sequence, and this single base difference is known as SNP, that is single nucleotide polymorphism. One SNP occurs at every approx 1000 base pair of human genome, and about 85% of all differences in human DNA are due to SNP, that means single base pair difference. That is the human chromosome 1, the genetic map of first chromosome. Now, what was the HGP, or what was the Human Genome Project? The Human Genome Project was an international research effort to determine the sequence of human genome and identify the genes that it contains. The US Human Genome Project is a 13-year effort which is coordinated by the Department of Energy, that is DOE, and National Institute of Health. Okay, in this given image you can see proteins are formed from the genes. Okay, after that there are some milestones from which you can get the information like birth of HGP and when it has completed all this information. Now, the goals of Human Genome Project. Goals basically include, first, with the help of Human Genome Project, we can identify all the genes which is present in human DNA. Second, to develop a genetic linkage map of human genome. Third, so that we can obtain a physical map of human genome. Physical basically include the how much distance is there between the gene. Next is to develop technology for the management of human genome information, to know the functions of the gene and determine the sequence of 3 billion chemical base pair that make up human DNA. Seventh one, store this information in public databases. Next is develop tools for data analysis. And the last goal is transfer related technologies to the private sectors. Now, there are some issues of concern. Some are ethical, legal and social issues of Human Genome Project. First is fairness in the use of genetic information. Second, privacy and confidentiality of genetic information. These are the issues. Next, psychological impact and stigmatizations and discrimination. Next is reproductive issues also there. Clinical issues is also there. And uncertainty associated with gene tests for susceptibilities and complex conditions. Next, fairness in access to advanced genomic technologies. Conceptual and philosophical implications, health and environmental issues, commercialization of product, education, standard and quality control, and the last one, an important one is also, that is patent issue. Okay, so these are the issues of concern. After that, there are some future challenges what we still don't know about human genome. So first is the gene number, exact location and the function. Second is gene regulation. Third, chromosomal structure and organizations. And there are some non-coding parts also present there. And amount, distribution, information, content and function of these non-coding DNA. Next is the coordination of gene expression, protein synthesis, proteosomes and post-translation event that we still don't know. Predicted versus experimentally determined gene function, evolutionary conservations among organisms, disease predictions based on gene sequence variation, genes involved in complex trait and multi-genes diseases, and developmental genetics, genomics. These are some challenges that we are still facing. Okay, so up to that slide, if you have any doubt then you can ask me.